Okay, welcome back everyone. It's a particular pleasure for me to introduce my colleague in Population Health Sciences at the University of Bristol, George Davey Smith. We were happy to host this conference face-to-face in Bristol, so it feels particularly apposite that our final presentation of the meeting is Bristol-based, albeit virtually. George Davey Smith is Professor of Clinical Epidemiology and Director of the MRC Integrative Epidemiology Unit. George is a world-renowned clinical epidemiologist and prolific author, so it's difficult to briefly summarise his enormous contribution to the field. But I'll try by saying that his work has particularly focused on methods for improving causal inference in studies of disease etiology and disease prevention. As you can see from the title of his presentation, George likes to take the long view and to use history to cast an interesting and thought-provoking light on contemporary epidemiological and public health issues. So you're in for a treat. There will be some time for questions at the end, so please put your questions in the chat and I will feed them to George afterwards. So at the risk of sounding like a NAF TV anchor, George, over to you.
Thanks. Cheers. Thank you. So I'll just share my screen. I've now just screwed up my ability to do that after having practiced it for so long. Sorry. Really sorry. There. There we go. Sorry about that. Hopefully that's visible and in presentation mode. Yeah, so this is just reflecting on experimental epidemiology and immune ecology. What I'm going to do is very briefly discuss what experimental epidemiology was, what it achieved, and the human analogies of experimental epidemiology, which is animal study based, and then immune ecology and its relevance for implications for today. So before experimental epidemiology, which arose in about 1920, had been the post-germ theory years of success with vaccinations, ways of dealing with typhoid and cholera and other infectious diseases through public health interventions. There were informative failures like the cholera outbreak in Hamburg, 1892, but that could be dealt with by basically improving on the methods which were being used. And Major Greenwood, the most important British epidemiologist of the first half of the 20th century, he said in Nature in 1916 that epidemiology was going to be elevated by mathematics, and it's going to be moved from its humiliating position as a plaything of bacteriologists and public health officials to infectious disease modelling. By infectious disease modelling by Ronald Ross, Brownlee and others, we're going to make it finally a distinct science. Then came World War I, but in particular what came was the Spanish flu, which of course shouldn't be called Spanish flu, Kansas flu maybe, which epidemiologists and public health people just had nothing to give to, had no way of conceptualizing it or dealing with it. Greenwood wrote the 400-page report for the Ministry of Health on the influenza outbreak. And the bacteriologist and his colleague in experimental epidemiology, Topley, Greenwood said in his obituary, was led to the conclusion that orthodox epidemiology was little better than a blend of folklore and mythology, only to be transformed into a science by turning it into a branch of experimental biology. Greenwood, when he wrote his textbook, one of the first textbooks of epidemiology in 1935, still reflecting back on this, was saying that he ended on discussing influenza, where we have our failure is complete. We have still nothing to say about that. So others in other areas, like in poetry, this post First World War situation led to writing the wasteland in Eliot's case and the fragments that he shored against his ruins. For epidemiology, these fragments that were shored up by introducing experimental animal studies as ways of becoming transforming into a proper science. There were branches in the US and the Rockefeller Institute in Germany, in Berlin, and in Melbourne, but I'm going to talk about the UK work. The work but was based upon the standard notion of the disease triangle, of thinking about the agent, thinking about the environment, and thinking about the host, or the soil, seed, and sower. For the 20 years of work by Topley, Greenwood, and then junior statistician Bradford Hill and Wilson, who was a laboratory scientist, summarised in this report, but just filled probably hundreds or thousands of pages of journal articles. So the studies were in mice colonies, and this is oversimplifying the large number of designs, but the major designs would be you would start with an uninfected colony and you would just carry on adding susceptible mice in your control conditions. This was with no infectious agent being introduced, and then you would sort of count the deaths and replenish them. In a second experiment, you would add infected mice to the colony and at the start, after a running period, and then regularly add more susceptible mice to keep the population level up and count the dead. In a third experiment, you would mix infectable and susceptible mice through adding mice who'd had prior exposure and seeing what happened to them when they're added to the colony in which infection was spreading. So looking at how mice, and then in the fourth study, rather than adding mice who'd been infected, you would add vaccinated mice and see how they fared. So these were the basic experimental setups. Many infections were studied. Ectromelia or mousepox was the viral infection, which was studied in detail. Now, after 20 years of that work and work carried out in the US and in Melbourne in particular, and work carried on in Melbourne until the 1950s, the main conclusions were firstly that there was just very large inter-study variation. You were doing precisely the same study again, you thought, and yet the results would turn out to be very different. In the 1936 report, they discuss, in one experiment, they discussed the very unfortunate history of the controls, was that for a reason that they couldn't work out, suddenly they had very high mortality rates in supposedly uninfected populations. So stochasticity was a major thing. Another conclusion was the major importance of infectious dose. So the actual dose of infectious agent that was received by the animals would have a major influence on their clinical course. A second conclusion with respect to the mousepox was that attenuated live virus immunization was protective, but that the immunized animals could still carry unspread virus. But when the immunized animals or survivors of infection did become infected, they were often asymptomatic, and they didn't develop, even if they did develop symptomatic, well, it's detectable disease, it was not serious, and mortality rates were very low. That there was generally a constant strain within epidemics, but some changes in strain occurred between them. This was a key issue at the time, because one of the theories, Brownlee's theory of how a single epidemic pattern would go, was that the actual infecting agent changed, and that is what led to the rise and fall of the epidemic curve. So they excluded that, but the strain would change between the epidemics. And they also discussed how the pre-existing immunity accommodates to the changing strain. So if the animals who had immunity, either from vaccination or from infection with the previous strain, that they would accommodate the changing strain and not generally become seriously ill or die. Greenwood made many conclusions of considerable relevance to epidemiology, we would recognize them today. In modern epidemiology, you can't observe counterfactuals, as he'd say. Any experiment you can only randomize. You can only not randomize. You can only give the mice one thing. That he discussed the important contribution of what you can't even hope to measure. How many of the interactions in these colonies, even though these colonies were so tightly controlled, couldn't be measured, and the massively underappreciated role just of chance or stochastic events. He introduced the notion, obvious in infectious disease, but equally relevant to other conditions, that the properties of a group are often, in fact, usually different from the sum of the properties of the individuals, or even the subgroups making up the group. And he also discussed how, when studying infectious disease, your study sample was the group, was not the individual. He talked about how 1,000 mice would make up what would produce one measurement. It would be one epidemic, and that over 20 years they sacrificed hundreds of thousands of mice, and even with that sample, statistically, they had little power. The role of chance, I think, is very beautifully demonstrated in modern data from SARS-CoV-2 and this extraordinary sequencing effort that's available. Now, even with, of course, the non-systematic sampling of the sequence data one has, you have this beautiful illustration of when looking at the transmission size lineages that you have, that you can actually put together. You have a very large number of very small transition lineages, and then you have a large number of over 30, where you've found over 30 cases. In between, there's the chance of them dying out, but if they get to above a certain number, they're likely to go forward and not be eliminated stochastically. So this is a rather beautiful demonstration of that with data. So that was the mice. Now, the Medical Research Council's other investment was in human studies, in particular run by S.F. Dudley, who was with the Royal Navy Medical Service, reviewing his 1926 MRC report. One of the reviewers said, there are some communities sufficiently isolated and under control to have much the same value for purposes of study as a group of animals under experimental conditions. This was Dudley's report on the spread of droplet infection in semi-isolated communities. This is one of them. This is the HMS Impregnable, which is a Royal Navy school. This was the school for the poor kids going to the Royal Navy. As you might imagine, it was not impregnable to infection. One of the second communities, there was the London Foundling Hospital that was studied. The other was the Greenwich Hospital, which was the posher Royal Navy school. This is where Dudley's work on diphtheria in particular was done. He put the human herd Greenwich Hospital schoolboys at dinner under the photo that was the facsimile that appeared with this 1934 report. And it was said that his use of the term about the experimental herd was raised in the House of Commons because Dudley was making these helpless school kids subjects of experiments. But I went back to Hansard, and it wasn't actually in the way. It didn't actually happen. It's a good story, but it didn't happen in that way. So the findings from Dudley's studies, firstly, was that the germ is not the disease, that there were many carriers who would carry an infection that would not be detected. There was a large amount of latent immunization that had occurred, but it wasn't possible to date when it had occurred and precisely how it had occurred. He investigated the influence of immunity and whether induced by infection or immunization would reduce infection risk, but obviously not abolish it for many conditions. He studied different conditions, but would reduce the severity of disease and the mortality risk, and also the infectiousness of those infected. He was particularly interested in studying the maintenance of population and individual immunity through reinfection, and in particular the importance of age of infection, which I'll come back to. Dudley is an extraordinary polymath and writer. One of his papers on herds and immunity. He had great advice for public health practitioners. He pointed out that the announcement that some famous film star or pugilist uses a quack medicine seems a far better advertisement than an endorsement by the whole College of Physicians. Public health administrators should make more use of this peculiar quality of herd instinct and subsidize some notorious criminal or other herd hero to broadcast genuine health propaganda. I guess as we have these press conferences from Boris Johnson, we sort of get that. He wrote about the human adaptation to the parasitic environment and reviewed in very considerable detail outbreaks of infections in populations who lacked immunity and the very serious consequences of that, and ultimately came to formulate this whole notion of ecological epidemiology. He was definitely the first person to formulate that so clearly. And in his paper on ecological epidemiology, or the ecological principles in epidemiology, he discussed Burnett's law, which he very generously named after Sir Macfarlane Burnet, the Nobel Prize-winning virologist, who had studied many infections and looked at the role of early life infection, which would not produce serious clinical disease. This would be the case in yellow fever, in polio, and in malaria to an extent. But then, if these children were not infected and became first infected later, the disease would be more serious. So this notion that when a new virus arrives, is ubiquitous. The young children who become infected do not get a serious disease. It's the previously unexposed adults, and particularly older adults, who become sick. I'm now going to move on to the concepts that were put together by the experimental epidemiology movement and by the work by Dudley and others in closed communities. The first is that which is considered to have the unfortunate term of herd immunity, which Topley introduced. Topley and Wilson, who were working during the reports of the early experimental epidemiology studies, this notion of the immunity within a population and how the sum of the immunity for the individuals is greater than has effects which are beyond that of individual level of effects. Topley discussed that they're indeed applying to the herd the same test of immunity as he applies to the individual, must be able to withstand exposure to risk, i.e. for the herd. With herds, as with individuals, there are of course all gradations between complete immunity and complete susceptibility, but it is a relative thing. So you're looking at a relative phenomenon, not that it's completely protected if the infection is introduced to the community or the individual, but that they have relative protection. In fact, even though the Topley's 1923 paper is where the source of herd immunity, the herd immunity concept, has generally been referred to, it did actually exist with respect to herds when looking at abortion disease in cattle, and had been, although not in the mainly academic literature, this was in the practical literature when trying to give advice to the dairy farmers. So, but perhaps it's unfortunate, and with the name herd immunity raising antibodies, it's unfortunate that we didn't stick to the mice, because the collective name for mice is a mischief of mice. So then we just call it mischief immunity, which would maybe not have the same, lead to the same level of upset. For the second concept they discussed, they had an even more unfortunate term than our current term. Currently, we refer to harvesting, the notion of mortality displacement, that people, particularly very elderly, very sick people with pre-existing conditions, have elevated mortality, and that diseases like SARS-CoV-2 are clearly very good at leading to terrible outcomes amongst people who are with pre-existing conditions. They actually referred to it as mortuary selection, which I guess is an even grimmer name, the notion that the disease would select out the more susceptible. They also discussed the role that natural immunisation may play in increasing the average resistance displayed by surviving mice. So these two forces. These are the data for the sort of first wave, I guess, when we with the deaths attributed to COVID and excess deaths not including COVID-19, with little evidence of any sort of substantial reduction in excess deaths or in deaths not caused by COVID-19. And of course, in the second wave, for quite the last several weeks, there's now an overall negative excess deaths, which does suggest that there has been some mortality displacement. They were particularly exercised by the notion of virgin soil epidemics, when infection arrived in populations without immunity. And this is, again, something that might be of concern now, given that because of the restrictions, etc., infections like the common human coronaviruses, these are data from the US, have not risen to normal levels. Influenza rates are low, and other generally minor infections have been low. And here, there have been reported in the past, there have been quite a lot of single case study reports of outbreaks of the common human coronaviruses, the seasonal coronaviruses, leading to severe outcomes amongst groups who might have lost immunity because they had not had infection just by chance. Infection hadn't been introduced into that care home, say, for 2 or 3 years, and when the immunity which stops serious illness had dissipated. Now, the final concept that Dudley and Burnett and others discussed was this notion of immune ecology, which is that the immunity level in the population is shaped by artificial immunization and by circulating infection. And the notion that as for example, the viral strain changes, the immunity will adapt to that and expand and become broader in the way that, for example, influenza, immunity to influenza broadens with age, with repeat infections, and gives greater and greater coverage. So thinking about implications for population immunity, which is created both by artificial immunization and by some levels of infection in our population, I just tried to put together a few things that I think need to be known in terms of working out the strategies which will be optimal in terms of reducing severe morbidity and mortality. So obviously we need to know the changes which occur in the virus, just due to drift, and changes which occur due to selection against a background of natural infection, or selection against a background of vaccination-induced immunity. So how is the selection of potentially more transmissible, more virulent strains influenced by the level of natural and the level of vaccine-induced immunity? And clearly, we have now a background of mixed immunity, as the ONS surveys, which I participate in, clearly demonstrate. And then, in terms of what we need to know in terms of disease ecology, is how that immunity is constructed by vaccination and then by circulation of virus, and how with the changes in viral type, how that accommodates, how the immunity accommodates that with respect to the health consequences. So the matching of immunity to current viral types, and how that could be done either by constant artificial immunization with change of the form of vaccine, yearly, 2 yearly for the whole population, or thinking about segmented approaches. And then, so that's it. I'd just like to thank people who helped me with the PowerPoints, and that's it. I'll stop sharing.
Thanks very much indeed, George. So we've got a little bit of time for questions. There's nothing in the chat yet, but please get busy there if you've got questions for George. But in the meantime, George, I'll perhaps start by asking you a question. I wonder, you've sort of focused on mortality, but does any of the historical work that you've looked at offer any insights in terms of the phenomena of long COVID?
No, I mean, clearly in the sort of animal studies, not, and even, I mean, so the populations Dudley looked at were under, constant surveillance, and were constantly being looked at. But there really wasn't any. There really wasn't any discussion of longer-term consequences. I mean, it was acute illness which either led to, in those days, quite reasonable mortality from diphtheria and other infections. I mean, there clearly was discussion, although not in experimental epidemiology literature, about consequences of flu. In particular, the 1889 to 1894 flu pandemic, when the whole concept of neurasthenia developed alongside what were seen as the long-term consequences of that pandemic. Even though, of course, the mortality consequences were much lower than in 1918, it had this very particular imprint of whole new categories of disease being constructed around it. It's extraordinary history.
Great.
Mark Honningsbaum's book about the 1889 to 94 flu pandemic is highly recommended.
Great. Okay, George, I've got a question here from John Frank, who says, fabulous talk. Do you believe that undocumented cross-relativity of immunity from prior or other coronavirus infections helps to explain the very high unexplained variance across countries, continents, e.g. South Africa now, in COVID impact?
That's a great question, John. So with respect to other, i.e. just means the 4 seasonal coronavirus infections, I think that is unlikely, partly, particularly because there appears to be rather limited cross-reactive immunity between the 4 species. So the studies which have been done, suggests that if you have one of the 4 types, then that provides rather little protection against when the next one comes along, if any protection at all. So the situation with South Africa would not be thinking about other coronavirus infections. I mean, it would be thinking about infections which stimulate T-cell immunity. So you'd be thinking in particular of tuberculosis, other mycobacteria, infections, and that even though the amount of very strong evidence on that, I think, is limited. That is plausible in terms of mechanism, and it's more plausible than there being an important effect of seasonal coronavirus, prior seasonal coronavirus infection.
Great, thanks, George. I'm just looking in the chat. I'm going to ask you another question. So, which is perhaps a bit unfair, but so sort of based on this work, George, what would be the sort of key advice that you would give to Chris Whitty right now? Because we're public health folks, so.
So I think I wouldn't dare to give Chris Whitty advice, but I think one should when considering what the vaccination strategies should be, I think what one should consider and model the effects on morbidity of immunity responding to a changing virus. If say, infections would spread through kids, which is how this would presumably move to become as a seasonal coronavirus. It'd be very unlikely if it'd be very odd if it didn't. How possible it is to mimic that as well with changing with manufacturing new vaccines every year. I mean, it's not as though it's such a crucial issue that if you start vaccinating children, it doesn't mean you have to carry on forever vaccinating children. But at least I think that should be considered. I mean, it's obvious that one would continue vaccinating the high risk populations, as with flu vaccine. But at least one should. I think at least different strategies should be considered.
Great. Okay, George. Well, look, thanks very much indeed for your fascinating presentation. I think that's really brings us to the end of this session.